So Greg joined us about 18 months ago from the University of Queensland in Australia, where he'd spent 18 months of extended sabbatical, having spent 10 years before that at North Carolina State University, where he established himself as a quantitative genomicist, mainly working on Drosophila. The last few years he switched to studies of human genetics, and he came to Georgia Tech to establish a program and a center for integrative genomics here, a program built around the idea that systems biology can be used to sort of bridge a gap between epidemiological things at the population level and the sort of cell and molecular biology and engineering that we do here at Georgia Tech. And today he's going to talk about that interface. Oh, thanks very much, Bob. So it's a pleasure to be here. So as Bob mentioned, so I work on really genomic epidemiology these days. So what I'm interested in is the consequences of all different environmental influences and how they interact with the genome to influence phenotypic diversity and disease susceptibility. So when people move from rural areas to small towns or to big cities, we know that that actually impacts disease susceptibility in many ways, whether it's schizophrenia or metabolic disease. And we know that events that happen during a person's life, such as economic crashes, impact their health. We know that parental effects, and increasingly we see that grandparental effects, have an influence on health. And of course, we know that there's an enormous component of genetic susceptibility. And in the last few years, there's been this incredible revolution in human genetics where we have the capacity now to probe the entire genome, and pretty soon we'll have the capacity to sequence entire genomes for $1,000 or so. To really get at the genetic basis of variation. But of course, as you're all probably aware, the findings from that are a mixed bag, because we have this microscope that gets us in now to individual variants, and there have been thousands of variants found for hundreds of traits. And it's an astonishing advance, but the cup half empty view is that it's only explaining 10 or 15% of the genetic variants, even with samples of 50,000 people. Okay? So there's a lot missing. But the other thing that's missing is the environment. Okay? So we know that genes interact with the environment, and my perception is that if we're going to understand that, we need more functional sort of assays that sort of don't just go from genotype to disease risk, but open up that black box and go from genotype to gene expression to protein abundance to metabolite abundance, and so forth. So that's what we're going to try to do in the center here, is add those components. Okay. So I wrote a book a couple of years ago called It Takes a Genome, built around the idea that there's been a dramatic shift in our environment in the last 2 generations, or in fact the last generation in many parts of the world. And there's been dramatic shifts in our genetics over the course of the last 100,000 years or so as we've migrated around the world. And as a consequence of that, the human genome is not in equilibrium. And my conception is that disease is arising because of that imbalance between our genetic heritage and our modern world. So we're seeing this demographic shift, if you like, of disease susceptibility from being infectious diseases and diseases of malnutrition to globally now the diseases of the affluent: cancer, diabetes, asthma, depression, cardiovascular disease, and so forth. And the real question is, why is that? Because our genomes haven't changed that much in the last generation or so, but our risk certainly has. So what I'll do today is start by presenting, the first half of the talk will be 2 sets of studies we've done that have got us into the human genetics. And one approach, the first study is what I call geographical genomics of this human transition in lifestyle. And I'll discuss a signature of the effect of environment and lifestyle on our gene expression profiles in peripheral blood. And then I'll describe a genome-wide association study for gene expression in the blood that we did in a sample there. And then the second study, this is stopped, there we go, is motivated by this concept of thrifty phenotype, which is the notion that maternal health is impacting the child's health already at birth. And so we use gene expression profiling to get at that. And then I'll spend the last 15 or 20 minutes talking about integrative genomics, what we're doing at Georgia Tech, and how I see what we're doing interfacing across IBB and the university as a whole. So Youssef Irakhdor here was a graduate student in my lab who came on a Fulbright fellowship, and I talked him into going back to his home country, Morocco, and doing this study. And he took it and ran with it and did an absolutely tremendous job, not just in my view, but a lot of views. He's won several awards for this work. And it goes like this, that we decided that we wanted to sample people who were living different lifestyles. And the bottom line is the idea that our disease risk is going to be different if we're living sort of traditional human lifestyles, whether we're desert nomads or Bedouins, or if we're living in very small rural mountain communities, or if we're actually living in modern cities, in this case in the slum of this city of Agadir here on the Atlantic coast of Morocco. So I like to sort of just since this is early in the morning and we're all waking up, I'll just use a, oops, how do I go back? Just use this Google Earth flyover video to sort of illustrate Morocco, which if you get a chance, I recommend going there because it's one of the most beautiful countries on the planet. So we went in and we started in Agadir, which is this city here. So there's the beach, which is full of English tourists getting sunburnt. And we go about 2 or 3 miles north past a concrete smelter there that you see. And then you have this dense urban sort of low-income community here by the ocean with fantastic views, but you know, very bad respiratory problems, bad air pollution, very crowded. That's through downtown Agadir, and then we come to Chira, which is another sort of low-income and dense region next to the airport here, where we sampled. And the communities in both sides of Agadir are a mixture of Arabs and Berbers, or formerly Amazigh people. So the Arabs came into Morocco in the Middle Ages, so what, 6 or 7 centuries ago, and they typically live in the desert valleys. So this is another city, Tiznit. That's in fact about 50 meters from where Youssef grew up. So they live in the dry valleys, and are typically fairly segregated from the Boutroch villages, from the Berber villages who are living higher up into the hills of the Low Atlas Mountains. So that's Ihram, where Youssef spent his childhoods in the summer. So there's no electricity whatsoever, just getting it now. Running water had to come from nearby streams and so forth. This is the first village we sampled from, Set Nabor. It took him 6 hours on a donkey to get up there, so that's fairly remote. And then the second village is Boutroch that we sampled from. Is also a Berber village, 100% Berber. There is a road, as you'll see, going through on Google Earth here, but that went in literally 2 years ago. So again, a very isolated village. Then the other place is about 12 hours away by four-wheel drive, way out into the middle of the Sahara Desert, where we were lucky enough that we were able to come across about a dozen small family groups that sort of move every 3 weeks, move their tents. So you saw one in that slide a couple of minutes ago. And incredibly friendly people, you know, wanted to share a goat with us for lunch, even though they only have, you know, 5 or 6. I mean, it's just amazing, amazing experience. So that's Morocco. And actually, I was so excited I was able to send a picture back to my lab from an hour out into the desert from right there. Who would have thought that? You go to the middle of Manhattan, you can't get service on a cell phone at all, but in the middle of the Sahara you can. So the question we asked was then, how different is gene expression between these people? And we were sort of thinking that maybe we'd find half dozen genes or a few dozen genes that were significant genome-wide. And we teamed up with a guy called John Story, who's one of the leading statisticians working on analysis and normalization of gene expression data, and he used something called surrogate variable analysis to really do this properly. But it doesn't really matter. No matter how you look at it, you see that there are these different groupings. So it turns out that the villages, the remote villages, are the most different. So you can see all of those remote villages here in blue are clustering together with a couple of exceptions. And then pretty much the Bedouin living out in the desert and moving around, the nomads moving every few weeks, or another group. There's a subgroup within here which is in fact some of the women Bedouin compared to the men, and then they're the people in Agadir. So 3 different groupings. So it immediately says that your lifestyle is having a major impact on your gene expression profiles. And how many genes is it? Well, we do the analysis of variance on it. It turns out that in this analysis about 1/3 of our transcriptome is different according to where you're growing up. And so we adjust this for ethnicity and so forth. And that's at genome-wide significance levels, which means that we're looking at 10 to the minus 6, 10 to the minus 7 p-values on those genes. Okay? So it's not just a few genes, it's a very big effect. And it's also not random. So if you go to things like ingenuity pathway analysis, and they've annotated the genome with respect to sort of different disease classes and other types of gene ontology, you see that the biggest difference is in fact genes classified as being involved in respiratory disease, separating the urban versus the rural samples, and that made perfect sense because that urban sample is suffering from a very high level of respiratory illness. We were wondering at that time whether this might also engage with epigenetic effects, so we just did a methylation profiling using at the time the cancer methylation panel that Illumina had. So this was only about 1,500 genes at that time that had just CPG islands that you can do this sort of bisulfite conversion, then it becomes a genotyping chip. But it's quantitative and it allows you to estimate levels of methylation in front of these 1,500 genes. And our positive control is in fact a sex effect. So there were about 150 genes that were different between females and males, and they turned out that every one of those was on the X chromosome, as you'd expect, because you have differential methylation of one of the X chromosomes. But if you look at the lifestyle effect, there were only about 24 genes that were differentially methylated, whereas, as I said, there were several thousand genes differentially expressed. So it's not obvious that methylation per se, at least at this low-level resolution, is responsible for those differences. Although I have to say that one of these genes is in fact the transcription factor ELK1, which is one of the major transcription regulatory factors in the peripheral blood. And in fact, it turns out that targets of ELK1 are enriched among genes that are differentially expressed. So it's possible that a few epigenetic changes could actually ramify and affect a large amount of the transcriptome. I doubt that that's the case, but it's certainly possible. But the bottom line there is that epigenetic modification by methylation, at least at this level of resolution, is only accounting for a small fraction of the expression divergence between those localities. Doesn't mean that histone modification, microRNA changes, and so forth aren't also involved, but it would take a lot more work to sort of follow that link. So we decided to follow up and with a much larger sample. So that first study was only a few dozen people from each of the locations. So we went back about a year and a half later, and decided to get 200 people because it had just appeared that you can do genome-wide association studies for gene expression on 200, at least from lymphoblast cell lines that people had looked at. So we gathered about 100 samples from both sides of Agadir, Anza and Chira, and about 50 from this Arab village of Igram, and about 50 from this Amazigh village of Boutroch. Now there are some Amazigh living in Igram as well, and most of the men in Igram actually commute into Tiznit to work, so there are lifestyle differences there. So we got peripheral blood samples, which we immediately are able to fractionate the leukocytes out, so we have a size fractionation filter that removes the platelets and the red blood cells. That goes straight into RNA later on the filter, so it's preserved. We ship it back to the States, and David Goldstein's group at Duke University ran through the gene expression profiling for us on these human HT12 arrays, which have 48,000 different transcript probes representing all of the genes, all of the RefSec genes, plus a number of others, with many of them with multiple probes. And then we also did the genome-wide genotyping on these with the 610 quad arrays at that time, so 600,000, which after quality control boils down to about 500,000 SNPs throughout the genome that we can genotype. So the question is, how does variation in the genotypes affect variation in the transcripts, given the ethnic variation and the geographical variation that we're seeing? So when you do these studies, you have to control for ethnicity. So these are what we call principal components analysis of the genotypes now. So we're looking at the genotypic structure. So what impact does your genetic diversity have on where you cluster relative to other people? So the first component of genotypic variation actually separated about a dozen people out from the other 200 or so in the study. So these people, and these three individuals here, I guess I've been politically incorrect because I labeled them as black, but they actually are sub-Saharan Africans. And the others were self-reporting as having some sub-Saharan ancestry there. Okay? So we figured that was probably what the axis was, and we threw in some Yoruban samples from the HapMap project, and sure enough, that axis is the sub-Saharan one. Okay? So there's just a small component, it's just a few percent of the diversity, but a few individuals who are able to contribute that. The remaining diversity is clearly along this second axis, so if we flip that on its side here, we've got the second axis, and then the third axis of variation is down here, and that's clearly separating Arabs from Berbers, or Amazigh. Okay, so these are the remote Berber village, these are the Arabs in the city of Agadir, these are the Berbers in the city of Agadir, and these are the Arabs in the city of Ihram. Okay, so we were expecting that you would actually have the Arab village at one extreme, the Berber village at the other, and then the city would be sort of admixed in the middle. But it turns out that probably the Arabs in the city are sort of more pure Arab, and you've had enough admixture with just a few individuals exchanged per generation over the last 20 or 30 generations since the Arabs moved into Morocco to actually bring the villages closer to the Berber genotype. Okay, so you've got a shift over along that axis, and then this axis here that's separating out the Ihram village is actually largely attributable to one portion of the first chromosome. So it's certainly not fixed differences, just minor allele frequency differences in that region, but they're enough to give a minor component of variation that looks like it's separating people. Okay, so we can actually take these measures of genotypic variation and we fit them into our subsequent models, and we control for population structure when we do our various statistical models of gene expression differences. And I actually work with statisticians at the SAS Institute, so Russ Wolfinger and Kelsey Miklaus on that, and then Peter Vischer at the University of Queensland in Australia is a sort of world expert on that methodology. Okay, so the first result is that we replicated the initial finding perfectly. So it turned out that the village of Boutroch, the Berber village, the very remote Berber village, was totally different from the others. In this case, almost half of the transcriptome, if you take a false discovery rate of 10%, is different compared to the city of Agadir, which is about 200,000 people, and that Arab village of Ihram. Okay? So it's a village, it's a rural village, but it's close enough to the city, I guess, that you get that lifestyle effect, shifting gene expression. We wanted to know whether lifestyle or ethnicity was having a bigger effect on transcription, and it turns out in this case that if you just look in Agadir, ethnicity and gender have almost no effect. So there's only a few dozen genes that are different by gender. There's maybe 5% different by ethnicity. It's just a minor component. Whereas if you look across the entire southern Morocco region, what's called the Souss, then you start being able to partition out ethnicity and location effects. So we think there's probably some combination of some contribution of ethnicity, but mainly it's probably geography, and that's confirmed in the next slide if we now look at the principal components of variation for gene expression. Okay, so that one a couple of slides ago was the principal components of variation for genotype. This is now gene expression. Okay? So people are differing along this axis across all populations, and in fact that axis we now think differentiates people in all populations we've looked at in Brisbane, and now in Atlanta we see variation along this axis. The second axis is somewhat specific to the Moroccan population so far, but what's intriguing is you get a very clear separation of these people from these people. And who are they? Well, these are the Berbers from that remote Berber village. So they're all quite different, men and women. These are the women from the Arab women from that Arab village. Okay? So the Arab women and the remote villages have got sort of one type of profile. And then the green are the residents of Agadir, the city. The open red circles here are the men from the Arab village. So we knew that they commuted, so that's not necessarily surprising they've got that one. But what's really weird is that the women, the Berber women from that Arab village, were clustering with this group as well. Okay? So we've got a very complex geographic, by ethnic, by gender differentiation there affecting their whole genome gene expression profiles. Okay? So that's a surprise. But it just brings home the fact that our expression profiles throughout the genome are modulated by our lifestyle. Our next question then was, are those lifestyle effects robust? Are the genetic effects on gene expression robust to these lifestyle effects? So in other words, do you see an interaction between your genotype and the environment in the way they regulate gene expression? Okay. I skipped. I meant to present this slide. So we can also do things like going in and saying, well, what sorts of genes are differentially expressed? So one of the groups of genes that's very differentially expressed are these small nucleolar RNA genes. So there's a whole family of about 20 of them, 15 or 16 of which are in the top 100 differentially expressed. Of these SNORDs, there's another family, SNORA, not differentially expressed. So I don't know what's going on with that. They're not in one place in the genome, so there's some coordinate regulation. They're annotated as having evolved a role in epigenetic modification, but that's about all I know. Oxidative phosphorylation, ribosomal proteins are certainly differentially expressed between the different places. I don't know if that reflects a differential rate of activation and maturation of immune cells, but it's certainly intriguing. So, since we have the genotypes, what we can do is do a genome-wide association study on gene expression variation. Okay, so a genome-wide association study is where you take genotypes, so in this case 500,000 of them, so 500,000 genotypes throughout the genome for every one of the participants in the study. And at each genotype you ask the question, is the different allele associated with the phenotype? So you can do this for diabetes, or you can do it for schizophrenia, or you can do it for height. Okay, so that's one trait at a time. And when you do that, you're looking for SNPs where you get a significant association at the level of 10 to the minus 8 or better, indicating genome-wide significance. So if you did, so for example, this was done, has been done for serum lipids, and they've found 100 variants genome-wide significant that affect serum lipid concentrations, and they explain about 15 or 20% of the genetic variation for serum lipids, about 30% of total variation. Okay. You can do it for diabetes. There are about 30 known genome-wide significant associations, again, that explain about 15-20% of the genetic variants. Okay. Those studies are done on upwards of 5,000 cases and controls. In fact, the serum lipids one is done on 150,000 people. Here we're looking at 200 people, okay, which you might think, well, that's not going to work, but in fact it works wonderfully. And this is what we call a Manhattan plot, where we're walking along the genome, so from the tip of the first chromosome down to the tip of the other tip of the 22nd chromosome. Okay, so each chromosome is a different color, and what you've got on the y-axis is called the negative logarithm of the p-value of the association. Okay, so a p-value of 10 to the minus 8 has a negative log p of 8, and so that's our cutoff. So 99.9% of the genotype associations are below that cutoff. But we have about 1,500 associations that exceed that threshold cutoff. And they're in 400 independent loci. Okay? So you see a big cluster here. That's actually on the 6th chromosome. That's in fact the MHC cluster. So there's a lot of associations there. So what it's saying is that genotypes in the MHC are regulating the expression of genes in the MHC. Okay, so we've got about 400 independent associations, and it turns out that many of those replicate associations that have been detected in, if you just look at lymphoblast cell lines that other people have done. This plot here is plotting the location of the SNP along the chromosomes against the location of the target gene that it's associated with. And what you see very clearly is that almost all of those associations are along the diagonal. And what that means is that the SNP is in the same location of the genome as the target gene. So in other words, these are variants in that gene that regulates its own expression. Okay, so they're promoter polymorphisms that are regulating gene expression. And the reason why this approach works so well is because it turns out that promoter variants will explain as much as 20%, in some cases up to 50 or 60% of the variation in transcript abundance for that gene. Okay? So genetic influences on transcription can be very, very large. They can explain 20 or 30% of it. But by the time you get to genetic influence on the disease itself, it's diluted out by the environment, by all of the complex interactions that are going on, and it's very rare for any genotype to have an influence of more than about 1% on the actual trait you get in the end. Okay? So that's why there's a lot of interest in doing the genetics at this level. So we can actually dissect genetic effects that regulate gene expression. We can see that they're large effects and study that much more easily than genetics of disease per se. Okay? And then there's about a dozen places where you have what are called trans associations. So these are SNPs in one chromosome affecting expression on another chromosome, but they're quite rare. And there's certainly some interesting cases there that actually capture known disease associations as well. Okay, but our question was, as I said, was how robust are those associations to the environment? And I could show you 380 of these plots showing that they're very robust. Okay, so what you're seeing here, this is the gene expression level normalized on some scale. So actually think about this scale as being, so it's a log-based 2 scale. So this is ranging over about a 2-fold difference. So minus a half to plus a half is about a 2-fold difference in expression. Okay, so this is the major homozygote. So that might be AA homozygotes. And these are the minor homozygotes. So these might be the people who are GG homozygotes. And then these are the people who are heterozygous. Okay? And so you're seeing that across the entire population, you see that these minor homozygotes have higher level of expression of this particular gene than the major. Okay? But the other thing you see here is that those lines, the red, blue, and green lines, are essentially parallel. So red is in fact Igram, so the Arab village, blue is Boutroch, the Berber village, and green is Agadir, the city. So those genotypic effects then are constant in the different environments. So we know the environment affects gene expression, but at least these cis-acting variants that affect gene expression are completely robust to that. And as I said, I could show you 380 of these slides. Here's a couple of cases where we actually see that Boutroch has differential expression relative to the other 2 locations, but nevertheless the genotypic effect is constant. This is a situation where it looks like there's crossing of line means. Okay, so in Agadir, the minor homozygotes have higher expression, but in fact there's only 2 of those, so it's a rare allele. So the allele frequency is less than 5%. There's only 2 individuals that are rare, and it's just as likely that those 2 were from these populations as the others. So that's probably just a statistical artifact. Okay? It may be real, but it's more likely a statistical artifact. Okay? So when we do the analysis, it turns out, sort of really contrary to my expectations, that there is no genotype by environment interaction at the level of gene expression. And that's a conundrum because, as I said at the beginning, I'm interested in this question of why it is that disease is increasing in the modern world. And at some level there's an interaction between genotype and environment giving rise to increased disease susceptibility. And yet when we look at the very molecular level of genotype affecting gene expression, we don't see it. So here's how I'm thinking about that. So if we just think of one example, okay? So I've just, this is a different example, but again, let's suppose it's an AAGG. Let's suppose that the people who are at a higher risk of some disease, diabetes for example, are the ones who have very low levels of gene expression. Okay? So you don't have enough transcript to do the job, and you're at higher risk of disease. Well, the only people who are really at high risk of disease are these ones down here. So it's the minor homozygotes in that population. Okay? So you can have an absence of interaction. So what we're seeing here, this is blue, this is Boutroch, okay? Low expression, intermediate expression, high expression. In all cases lower than you see in Igram and Agadir. But if what affects disease is gene expression, then the only ones at risk are these individuals here. So you could see an interaction between genotype and disease despite the absence of interaction between genotype and gene expression. Okay. So we're now sort of arguing and looking forward to sort of do this approach where we say, well, can we link the intermediate phenotypes to the disease through our genotype associations? So that's one study giving you a sort of sense of what we do. Here's another one motivated by this idea that maternal health during pregnancy in particular can influence their child's development. So the thrifty phenotype hypothesis is actually the observation that underweight mothers tend to have children who are at risk of early onset obesity and diabetes and other chronic diseases. And the argument is that if the mother's experiencing poor nutrition, then she'll reprogram the child's biochemistry to actually be able to rapidly assimilate nutrients. And then when the child is born in a world where there's a ready availability of nutrients, then they're going to put those into fat deposits, and you sort of simplistically speaking, get early onset diabetes and all the sequelae of that. I think more prevalent in modern society is the impact of obesity and overweight on disease risk. So we know that there's heritability of obesity, but we also know that we can tease that out because we can look at the mothers and the fathers' influences, and we actually know that maternal obesity during pregnancy is probably having a direct impact on likelihood of child obesity. So we said, can we detect that already at birth by looking at gene expression profiles? Now obviously the only tissue you can get from a newborn child is cord blood. Okay, so the cord blood is fetal in origin. So what we did is we set out to compare cord blood to maternal blood from matched mother-child pairs, where we had we wanted to have about 20 normal weight mothers, about 20 obese mothers, and about 20 gestational diabetic mothers. Well, for technical reasons, we didn't get that sample size, but we got enough to sort of start seeing some interesting effects. So, and that study was done by, you saw on the previous slide, Lizzie Mason. Some of you may remember she was here for 6 months when I first moved, and then Graham Trump was the OBGYN that we worked with. Okay. So in this case, we saw a whole new axis of variation that we haven't seen in general adult population. And so the first principal component of expression variation separates these individuals from these individuals. And we don't know a whole lot about what's different there. I can tell you that one of the differences is the ratio of CD4 to CD8 T lymphocytes. But that effect is at least as large as the effect of mother versus child. Okay, so this is the mothers. So the second principal component is separating the mothers from the children. Okay, and that's about 2,000 or 3,000 genes that are differentially expressed between mothers and child. That was known, replicated what other people had seen. What's new is this observation of these two classes of individuals, and we've got a large enough sample to start seeing this differentiation. If we go down further, and the other thing I should say is that if the mother is on this side, has low PC1 values, in every case her child had low PC1 values. So there's perfect transmission of whatever's going on in the mother to her child. We presume probably through cytokine signaling influencing both the maternal and the fetal gene expression. Now, neither of those axes are remotely associated with obesity or other obvious measures of health. But as we go down further, we see something interesting. So the third principal component axis is in fact equivalent to the first axis in the general adult population. But the fourth axis here is what I want to look at right now, because the blue spots, almost all of these, are the children born to obese mothers, and the pink spots are the profiles of the obese mothers themselves. So you get a very clear separation, which is significant at nominal sort of 0.01 level. And then the children born to gestational diabetic mothers are also on that side of the equation. So we're going to try to sort of replicate this here in Atlanta. But it's our first indication then that possibly there's an impact of maternal health directly on gene expression profiles for several hundred genes, which are in fact enriched for genes involved in central metabolism. We can do sort of network analyses of these. So this is a guy called Bruce Aronow has a program or a suite of software he's written called TopGene, which does sort of gene ontology analysis, but immediately outputs it into cytoscape and network analysis. So what you're seeing here is all of the differentially expressed genes between the type A and type B maternal or fetal profile with their network to known regulators. So these pink squares here are in fact transcription factors. So all of these genes are targets of these transcription factors. And then some of these, the darker purple ones, are microRNAs as well. So these are potential targets of those microRNAs. So you see a regulated response. So these are our candidates for mediating that difference. The brown ones here are all genes involved in chromosome modifications. I think there's a glucocorticoid receptor down here. There's, I think, various chromatin modifying factors here. But then what's interesting to us really is this yellow ones. These are all genes that are annotated as being responsive to methotrexate, which is a drug that mothers take to prevent spina bifida during birth. And it's also a drug that's taken in various other contexts. So, you know, what are the potential long-term consequences of that? We don't know, but I think it's pretty important to sort of consider that maybe people are differentiating their response to these drugs. And that's significant at a 10 to the minus 7 or 8 level of that association. So we can do those sort of network analyses. The other thing we can do is to hone in on specific pathways. Okay, so this is just a quick look at insulin signaling. And what we can do here is just cluster the people. So each column now is one of the individuals, and each row is a gene involved in insulin signaling. And you can see that some of the genes have high levels of expression, and other ones low levels of expression, and then they switch for different components of the pathway. So the green and the blue axes here are different with respect to this organization. So I'm actually teaming up with Melissa and Hang Lu to now start seeing if we can use microfluidic assays and other things to really hone in on particular pathways and link the variation we observe at the population level to real functional assays where you can go in and sort of knock out sort of what Melissa does, knocking out individual genes or doing specific assays, and really tie the variability at the population level to what individual genes are doing. And I'm certainly really keen to work with different people in engineering applications here and others to really see if we can add a functional link to what's going on. Okay, so we can measure gene expression profiles, metabolite profiles we're also doing. We can measure clinical covariates, but I think what we want to be able to do is also go in and measure actual functional aspects of these cell lines. I'm stretching this a little bit because this is actually not that pathway, but one of the components of variation also varied by location in Brisbane. So you can see here that the blue dots are all over the city, but there's an absence of red dots in the center of the city, and this is actually the sort of high income, high socioeconomic group region of the city. And we're interested to know whether that sort of thing also replicates here in Atlanta. So one of the things we can do is actually take people's zip codes and sort of do geographic analysis of that. So we think they're actually not just that you're seeing sort of rural villages versus cities, but we think there might be fine structure to variability across even large cities these days. And there's certainly some intriguing literature emerging about socioeconomic impacts on chronic disease and gene expression. Okay. So conclusions from that sort of work. Gene expression in human blood, we know it's highly heritable, but it's also extremely strongly affected by the environment. And what I'm interested in is dissecting the impacts of geography, gender, culture, and so forth on that. We're surprised not to see any genotype by environment interactions in these. But as I mentioned, what's intriguing is that additive effects at the expression level, so that's what we observe, might nevertheless translate into interaction effects at the disease level. And the idea is then that systems genetics is merging genetics with endophenotypes, such as gene expression, hormone levels, metabolite abundance, and so forth. And we're opening up this black box between genotype and disease. So instead of just doing genotypes with disease, we're doing genotypes with these endophenotypes. Okay. So that's all well and good. So it's sort of very sort of esoteric work or sort of basic biology. But what we want to do in the end is actually bring back these sorts of studies to disease. And as I mentioned at the beginning, I have an evolutionary background, so I've been thinking about these things in flies for a long time. And now I'm sort of trying to transport that way of thinking over to human genetics. And here's the question, so why is it that disease incidence is increasing? Okay. Well, we're all familiar with bell curves. In this case, what we're looking at is just susceptibility for some disease. Might be diabetes, might be schizophrenia. Okay. So most people are going to have an intermediate number of susceptibility alleles. Okay? Some people, by chance, are going to have more alleles increasing risks than not. Some people are going to have more alleles decreasing risk. And as we look throughout human history, systems evolve to make sure that susceptibility is decreased. That you minimize susceptibility to disease. Okay? So this is our point. You only have 1 or 2% above a threshold of susceptibility alleles giving rise to disease. And we now know that for most diseases, the number of alleles that are involved are in the hundreds, if not thousands. Okay? So it's a very small genetic effects, but nevertheless, systems have evolved to prevent disease. And the way most people think about what's happening now with the environment changing our susceptibility is that you're actually shifting the susceptibility allele count. Okay? Well, our genomes aren't evolving in the space of a few generations. There's not enough time for that. So somehow the interaction between genotypes and whatever the environment is increasing the number of those that are actually susceptibility alleles. Okay? So genetic effects which in the past were not sufficient to give rise to disease, in the modern environment are sufficient, and there are more of them, and so they're actually shifting your whole distribution over. The other possibility, which I get from my background in flies, is that in fact we're seeing something called decanalization. So instead of getting a global shift of the profile, we're getting an increase in variance of susceptibility allele effects. Okay? And the reason why you have 10 or 15% of people at risk now is because of this increase in variance, not so much as a global shift. So that's also something we're interested in. I won't carry on more about it for today's talk, but it sort of gives a background to why it is that we're now actually starting to focus on disease cohorts, which brings me to the what we're doing here and the role of the Center and the Predictive Health Institute in what's going on. So how many of you are aware that we actually have a Predictive Health Institute here at, well, Bob, thanks Bob. Just a few, all right? So it's a joint Georgia Tech Emory initiative focusing on health, okay, which is combining the strengths of both universities. So if we think about those, then we have enormous strength in bioengineering here or at both universities actually, but particularly the IBB, which is why I'm excited to sort of be part of this group. We have enormous strength in bioinformatics, particularly strong in comparative genomics, sequence analysis, and increasingly now in quantitative genetics. We have enormous strengths in things like healthcare economics, in database management, and so forth. And so one of the people I'm working with now is Nicoletta Serban in industrial systems engineering. I mean, it's just terrific to be able to integrate across so many disciplines and units here. And then, of course, Emory has a huge number of clinical cohorts. They have one of the strongest human genetics departments in the country. And they have access, or right there is the Children's Healthcare of Atlanta with its focus on pediatrics. Okay? So we can put these things together, and I think it's really underutilized at this point as to how much we're doing that. Now, one of the centerpieces of the Predictive Health Institute, so about $3 or $4 or $5 million were put into this by Emory 4 or 5 years ago. And what Ken Brigham has done is to set up this Center for the Health Discovery and Wellbeing, which is located up here at Midtown Hospital, up on Peachtree Street. And I'll tell you about that cohort in a minute. But as I said, I think there's unrealized potential for Georgia Tech involvement. And one of the other things, I'll just say it here, that I'd like to see happen is to have us bring in environmental biosensing. Because the places that are doing genome-wide association studies really seriously, you know, the Broads and the Wellcome Trust and others, are pumping hundreds of millions of dollars into that. I mean, they're really looking at 100,000 people at once. And we've got a cohort of 600. Okay? So we've got to be a little bit flexible about how we approach this, but we can do that. And one of the ways we can do it is by being very clever about engineering applications, and so doing things such as I hope you're doing with Melissa and Hang. But also I think the environmental component, people are just playing lip service to. We know the environment's important, but we don't go out and measure it. And so I'm really hoping that maybe there's some engineers in the audience here that we can do things like putting sensors in homes or attaching them to people's clothes and measuring how much exposure to PCBs and to other toxins in the environment. They've got lead and so forth, and we can integrate that into our studies. So the CHDWB cohort is about 600 Emory employees or friends of the university in the age range of about 25 to 75. Okay? So they're healthy Atlanta residents. Okay? But healthy is, you know, a relative term. So we've got about 30% obese in the study cohort, high risk of metabolic syndrome. So a lot of these people, in fact, most of them are more older than 40, and many will be coming into very high risk of cardiovascular illness, of diabetes, and so forth in the next 5 to 10 years. So it's really good timing to get this cohort. And what is happening is that they're coming in and getting a very, very intensive clinical profiling done, which takes about 5 or 6 hours of tests. So they're getting their blood cells drawn and getting complete blood cell counts and immune profiles. They're getting cardiovascular tests, they're getting psychological evaluation, bone mineral density, going through all sorts of scanners. And so that happens every 6 months. And then they're partnered with a health partner. Okay, so this is someone who talks to them on a regular basis about their diet, about their exercise regimen, about reducing stress in their lives. And the objective is to sort of change the way we do healthcare, put the focus on wellness. If you could reduce the rate of chronic disease, okay, so in America, what are the major diseases? It's cardiovascular, it's depression, it's diabetes, metabolic disease. If we can reduce those through the simple lifestyle intervention such as this, which is about $2,000 or $3,000 a year, the healthcare savings could be really quite substantial. Okay, so it's a pilot to see if that works, and what we've got the opportunity to do is to go in and do genomics on that. Okay. So with Dean Jones over at Emory, he's doing Fourier transform mass spectroscopy. So he's got 3,000 metabolites that he feels that he can robustly measure. So for the first time, we actually will have a cohort where we've got the whole genome genotypes, the metabolites, and the complete transcriptome we've done now. And we can put those things together and try to integrate those, of course, with all of this wonderful clinical data as well. So we've just got that data in, and we're just starting to go through it now. And then, as I said, we're actually working on functional assays as well. The most interesting finding that's come out so far is, in fact, not from this cohort, but it's with an affiliated one, which is Arshad Qayyumi's coronary artery disease cohort. So he's been bringing people into his clinic who get chest pain or more advanced coronary disease. And so we've profiled 200 of those and done the genotypes on those as well in parallel. So they're sort of a 10-year advanced cohort over the CHDWB one. And this is sort of new data. I'll come back to that. since I've introduced it, this is the sort of thing that I'm really excited about. So we've got a profile of about 500 genes. It's the second principal component of variation in this cohort, which is very strongly associated with death by myocardial infarction. So the people who actually in that cohort who subsequently died of a heart attack, if they have this profile here, okay, then they're pretty much unlikely to have died of a heart attack. Whereas if they have that profile here, then they've got much, much elevated risk of death by MI. So that's the sort of thing that we're really interested in going in now and looking at variability in that. And we want to know if this changes over time. So one of the other nice things about that CHDB cohort is we're getting profiles every 6 months of the people, and we'd like to see if those interventions can reduce that risk, take people out of this profile down into this sort of profile. And then, of course, we'd like to go in and sort of see what the genes are there. But the other thing that sort of strikes me in this cohort is just the repeatability of our measures. So this is triglycerides measured 3 times in each of 150 people. Okay? So there are some people who've got extremely high levels of triglycerides at enrollment and 6 months later. This is insulin. Same deal, extremely high levels. So one thing we can do now is actually go in and sequence those extreme individuals and see if we can find variants by sort of whole exome sequencing or others that might be responsible for these extreme effects. But even HDL, major predictor of heart disease, the repeatability of this is absolutely astonishing to me. The significance of that correlation, 3 repeat measures of 100 people, is about 10 to the minus 90. Okay? So people are there. So how able are we to get people from, in this case, low HDL is not good, up into this range by lifestyle interventions? That's one of our questions. Okay? And then we've got this rich cohort, and we can do things like asking how do genes affect simultaneously triglycerides and HDL. So here you see, we can repeat that measure that we know that people with higher triglycerides and low HDL are at higher risk, and so there they are. So that's what we want to be doing in that cohort. So other things we're doing, so there's a cystic fibrosis project with Niall McCarty here, and Arlene Stestenko, his collaborator. So we're asking the question, do airway and or peripheral blood neutrophils display a particular profile of oxidative or glycolytic stress that might differentiate the CF patients who go on to get diabetes from those who don't? Or does it differentiate all of these people with that chronic stress in the airway from normal people, and what can we learn about the stress in that system? We're working with Anne Mertens on a childhood leukemia survivor cohort that I just mentioned to some people in Barbara's lab earlier. So here the question is, does steroid and radiation therapy epigenetically modify gene expression, and is that related to the onset of chronic diseases like bone mineral density loss in that cohort, which is a very, very serious problem for early childhood cancer survivors? Again, the connection with the Children's Healthcare of Atlanta is fantastic. Just recently I've started working with Subra Kuthagasan, who's one of the world's leaders in pediatric Crohn's disease. So he's got a cohort there. He's leading an effort to assemble pediatric Crohn's data. So he's getting, so Crohn's disease is an inflammatory bowel disease, so he's getting bowel biopsies, he's getting the microbiome, microbial profiles, he's getting peripheral blood, all of these things, and we can put those together and ask whether a combination. In this case, our first question is, you know, we want to know whether genotypes can predict severity of disease and, in fact, the need for surgery. Okay, so it looks like that might be the case with some of the hits, but our thought is that a combination of genotypes with gene expression is actually going to improve that to a point where it becomes clinically relevant. So that's the sort of thing we're doing there. And then I think hopefully there's some people in the audience interested in stem cell work. So we actually have a grant in with Hang and Yang Sup to genome-wide association studies on variable iPS cell profiles, with the interest there of knowing how reprogramming is affecting individuality of profiles as we go ahead, particularly in the context of cardiovascular disease in this case. Okay. Oh, and then some of you may have seen the other sort of fun project I have on the side is a facial imaging project. So we're actually interested in the genetics of expression, of emotional expression. So we're looking at that in adolescents, but we're also looking at it in children. And we're going to ask the question whether we can find variants in particular genes that are associated with development of facial morphology. But going ahead, we're really interested in it as well as looking at the genetics of the emergence of the expression of emotions. You know, Darwin wrote The Expression of Emotion in Man and Animals. It was his third great treatise written 100 years ago, and there's almost no follow-up been done on that. So in the long term, that's actually something I'm very excited about. So, just to conclude then, so the Center for Integrative Genomics is a research cluster here that I'm putting together dedicated to the notion that systems genomics approaches can illuminate the molecular basis of disease susceptibility. But we're doing it in the context of my interest in the evolution of disease. And what I really want to do is to integrate what we're doing with bioengineering capacity and bioinformatics capacity that we have here to really make sure that Georgia Tech is playing a major role in the Predictive Health Institute as we move forward. And I think my last slide is an acknowledgement slide. This picture is actually from Youssef's cousin who won the UNESCO prize for the best photo this year. And so you see a little girl being taught to read in one of those villages that I mentioned. So Youssef, Sami Jadala was at the International Foundation for Conservation and Development of Wildlife, which is an institute dedicated to ecological research that supported us in Morocco. Russ, Wendy, and Kelsey at the SAS Institute, we've been long-time collaborators with them. David and Kevin at Duke, and then Peter Vischer is a great statistician at UQ. The Brisbane study was done actually, the profiling done in Christine Wells's group by Lizzie Mason. I mentioned Graham as the obstetrician, and I'm working with a whole lot of people here at Atlanta, so Ken, Shed, Niall, and others. Jinhee is the graduate student starting to work on this here, and then I also really want to acknowledge Dalia Arafat, who's my lab manager here. Thank you. This is Bob Goldberg.
Yeah, if you're going to do an impression, you got to get a haircut.
This is as far down as I go, man.
So that was great. Thank you. My question is, how did my gene expression change during Atlanta traffic this morning?
Well, I could tell you if you're a graduate student about to take an exam, it's going to change.
Questions?
Yes, very nice, Greg. I have a question about the thrifty phenotype. So it seems as though when women are malnourished during pregnancy, plus if they're overnourished during pregnancy, you still get these diabetes or cardiovascular phenotypes. Are they the same genes that change either way, or are they different pathways?
Yeah, that's what I'd like to know. I'd really like to know that. It's too small a cohort to get that. We only had about a half, 3 or 4. I mean, how malnourished can you be in Brisbane, Australia? You can certainly be, I think Brisbane won the award for the most obese city in the world last year, although you wouldn't notice in the city, most of those, there was absolutely no correlation with obesity in those profiles there, which surprised me. But yeah, we need a bigger study. So in fact, I'm really hoping to work with some of the pediatricians over at Emory to do a replication of this in Grady and Crawford Long. But it just takes quite a bit of effort to get the surgeons in line to be prepared to take those samples. Good question. No.
I wonder if there's an opportunity to do kind of an experiment where there's a major population shift, like say after Katrina when people just dispersed, changing their environment without changing their genotype, or in Haiti where people leave Haiti after the earthquake, to be able to try to track people down and get some kind of an experimental incident that separates their genotype from their environment because their environment.
Yeah, I think it'd be wonderful. I mean, my dream would be to sort of go around the world and do a map of expression profiles, how they vary by geography. We're getting a hint now in Atlanta that there might be a difference between ethnicities, but it's hard to tease that apart from socioeconomic things, so we want to be able to do that. The problem is that it's incredibly expensive to actually assemble those cohorts. It's just in personnel to go and find and get those. And you're not going to get funding to do this until you've got the studies in the freezer. Okay, so that's hard. So instead, I think the closest to that will be looking at migrant populations. So there's a very large, so Shed actually has a service where he has the Gujarati population here in Atlanta. So there's a very large North-South Asian population. Okay? A lot of interest in that. So they're actually have health fairs with those people. So we're starting to profile their blood because it's a population we can look at, we have clinical data on. And then compare that to what's going on in India. So I'm also working with Venkat Narayan, you may know at Emory in public health. So they've got a 4,000 person study going on in Calcutta. Because in India, so I mean, it's so fascinating. In India, you've got now some of the highest rates of diabetes in the world, but it's not associated with obesity. So in fact, one of the students gave a presentation in my group last week and sort of showed some genetic data, and she said, well, if I controlled for obesity, the signal went up. I said, well, that's really interesting. How did you define obesity? She said, BMI above 25. I was like, damn, I just lost 20 pounds and I'm at 25 and now you're telling me I'm still obese. But so that there's real, and then I'm looking in Fiji as well. I've got a student in Fiji doing things where you've got actually the Melanesian population so Fiji is about half Melanesian, half South Asian or Indian, because they were all brought into sugarcane plantations in the latter part of the 18th century, 18th century. Obesity rates in both are through the roof, 30%. But with totally different obesity profiles. You know, some South Pacific Islanders have had obesity for as long as we know. If you look at Captain Cook, the obesity was there, but they didn't have diabetes. So there it's probably associated with a shift in the sorts of oils that people cook with, or things like processed foods. So you know, the villagers will now go out to sort of buy roofs and improve their sort of living standards. They will actually sell their healthy foods, and then what have they got left for food? They have to buy processed canned stuff, and so their diet is terrible. So you've gone in one generation, this demographic transition, to very high levels of cardiovascular disease, hypertension, and diabetes. So we'd love to study things in those populations where we've got collaborating people.
Is that, you talked about not evolutionarily being able to catch up to some of the negative effects in our environment. Is that the reverse, that some of those developing countries, they have evolutionary conditions that now that they have better conditions, better nutrition and so forth, haven't caught up to that?
Yeah, so I look at it as that. So what we call an advance physiologically is not an advance. It's the other way around. Yeah.
I was curious about the work you were doing with the link between the genomes and our gene expression and the environment, and I was wondering if with more robust, more widespread experiments, you could begin to design or define an almost optimal human environment that we would be comfortable with, rather than waiting for big technology to influence our genome.
Man, you're sounding like Jim Neal. I mean, that's
Real estate.
real eugenic sort of stuff. The guy Jim Neal, so how many of you have heard of the thrifty genotype? Okay, so the thrifty genotype idea was that the reason we're getting diabetes is because there was humans during the recent evolutionary past went through massive periods of famine, so there would have been intense selection for the ability to store carbohydrates rapidly, and now that we've got ready availability of that, those genes that were good then are now bad for us. Okay, so it's sort of a classical fundamentalist Darwinian, neo-Darwinian paradigm, and it's almost certainly completely wrong. Okay, so all of the data now show, we know the 30 or so diabetes susceptibility loci, none of them have a signature of recent selection on them. Where am I going with this? Oh yeah, no. So anyway, so the guy who wrote the paper, so Jim Neal put this idea out there, and it was 40 years ago. So it was an interesting idea. Turns out to be wrong. But in the paper, he said, if it should turn out that in fact thrifty genotypes are true, then we need to do what we can to preserve them in the human genome. And you're sort of thinking, well, how are you going to do that? You're going to tell this person that you've got to mate with that? It's kind of scary. There was a lot of eugenics back then. So you're saying, can we create a perfect human environment? I think what we've got to do is realize that we're all genetically very different, and what's a good environment for you is not necessarily a good environment for me. And that's something we're actually, so I've also got a fly group, and what we're doing is growing flies on 4 different diets: a high-fat diet, a low-sugar diet, a high-sugar diet. And across the 150 lines we've looked at, there's absolutely no impact of diet on weight gain or other phenotypes. But the different diets do different lines do completely differently on different diets. So what's good for this line on so what this line does best on high fat, this line is going to do best on high sugar. And I think it's almost certainly true of us as well. And if you read Michael Pollan's work, I don't know, many of you know him, the guy who's written The Biology of Desire or In Defense of Food, he sort of makes that point that we've all got to, we can't reduce things too much to sort of saying that this is the optimal type. The take-home message is variability. Right? We're all variable, and what's good for you and good for me is not necessarily good for someone else. And we've all got to find our right place with respect to diet. So that'd be my answer there, in a roundabout way.
The colleague tells me we have another group coming in at 9:30, so we're going to bring this to a close. But let's thank Greg.